Hello everyone, my name is Sandra Deliard and I'm a postdoctoral fellow in Dr. Alessandro Gardini's lab at the Wistar Institute in Philadelphia, Pennsylvania, in the United States. Today I'll be talking about profiling the three-dimensional genomic networks essential for myeloid cell differentiation. The organization and dynamics of enhancer networks are key components in regulating myeloid cell differentiation. However, the underpinnings of this are not well studied. The CSF1R gene locus contains three enhancer regions, including the fire enhancer, which is bound by transcription factors such as EGR1 and is required for CSF1R activation and myeloid cell differentiation. However, studies showing fire enhancer's role in gene expression regulation have not been extensively reported. So here our goal is to determine the significance of the fire enhancer in regulating gene expression upon monocytic differentiation. We address this by using primary cells first, primary monocytes differentiated into macrophages. We then assess the DNA looping to the CSF1R locus, as well as identified any proteins that are enriched at these loop anchors. We also use CRISPR-Cas9 to target the fire enhancer region and see what deleting the fire enhancer in HL60 cells would do by comparing wild-type cells to mutant cells before and after differentiation, and looking at protein binding to the fire region, DNA looping to the CSF1R gene, as well as any transcriptional differences between the mutant and wild-type cells. First, looking at the primary cells, this is the CSF1R locus, and these are, we looked at binding of these transcriptional regulators at the locus. And we'll focus on the fire region here, and you'll notice that after differentiation, the fire enhancer becomes activated. And this is shown by increased EGR1 binding and increased H3K27 acetylation at the fire in macrophages. We next wanted to assess the DNA looping at this region. So we performed Hi-C in monocytes and macrophages. First, assessing the topological associated domains, or TADs, as shown here in these white triangles. And you see that there are a number of TADs in monocytes. However, after differentiation in macrophages, these TADs then become broken down into smaller regions. This could indicate that there is an increase in transcriptional activity in these regions. If we look further into the DNA looping to the CSF1R gene, we find that there is a ton of looping in monocytes. However, there is an increase in looping in macrophages in this region, as indicated by these dashes. If we look at the proteins that are enriched at these loop anchors, we find that in addition to the chromatin regulator CTCF and SMC1, EGR1, especially after differentiation, is enriched at these loop anchors. So this could indicate that EGR1 is playing an important role in monocyte differentiation through the regulation of the DNA looping to the CSF1R region. So we wanted to address this by next performing a CRISPR-Cas9 in HL60 cells and then comparing wild-type to mutant cells. And this is an agarose gel here just showing the fire region. In wild-type, you see there's one single band. However, in the mutants, you see that there are multiple bands indicating deletions in the fire region. If we look at CSF1R expression, we see that CSF1R expression is impaired in the mutant cells. And if we look at EGR1 binding, RNA Pol2, and H3K27 acetylation at the fire region, we notice that compared to the wild type after differentiation, there is a decrease in occupancy of all three of these factors at the fire region. Further looking at DNA looping in these cells, we see that in the wild type, after differentiation, there's a ton of looping that occurs. However, all of that looping is lost in mutant cells. If we look further into genes that are deregulated, or genes that are differentially expressed in the wild type cells, but not in the mutant cells, and they're highlighted here in gray, looking at one of the genes, IRGM, we do see that at the promoter that there is a ton of looping that occurs after differentiation in the wild type. However, all of those loops are now missing in the mutant cells. So this indicates that the looping could be a mechanism for regulating the expression of this particular gene. To conclude, we find that the fire enhancer, particularly the EGR1 binding site, is crucial for maintaining a network required for controlling the transcriptional changes required for differentiation. This illuminates the epigenetic mechanisms that drive the functional changes that occur upon differentiation, that also occur upon activating an inflammatory response upon infection. We further want to determine how depletion of EGR1 can affect the 3D conformation, as well as looking at the other enhancer regions and seeing what their roles are in maintaining this network. I'd like to thank the Gardini Lab, as well as the Wistar Genomics facility for the sequencing, as well as the Human Immunology Core at Penn for providing us the primary cells. And I'd like to thank you all for listening.